Yeah. So welcome everyone to our workshop that we're giving on FNIRS and EEG. We're happy to be here as a sponsor. I'm happy to give you a presentation and give you more insights in our devices, the technique of FNIRS, and how to combine FNIRS and EEG. We'll first start with introducing ourselves. So my name is Sam. As you can see, I've studied clinical human movement sciences. I have a background also in biomedical sciences, and I'm working as an application specialist at Artinis. My focus is mostly on NIRS. So we have functional near-infrared spectroscopy, and we have muscle NIRS. So I'm mostly focused on these two.
Yeah, and my name is Konstantinos. I am a junior software developer at Artinis, and I studied cognitive neuroscience, so similar background to yours, I think. And I focus more on EEG and high-density imaging.
So we're here on behalf of Artinis Medical Systems. I'll first tell you a bit more about the company, where we're located, and what we actually do. So we're a company based in Elst in the Netherlands. Probably you've never heard of it if you're not based in the Netherlands, but it's around the center, more to the east part of the Netherlands. So recently we also acquired an office in Oldenzaal. So that's over there. And we're a company that started in 2002. So we were the only NIRS company in Europe to develop and distribute NIRS devices. We have around 60 employees. Now I think it's even more closely to 70. We focus mostly on key markets as sports science and neuroscience. So we have devices for measuring brain, that's FNIRS and EEG. And we sell devices that uses HD EMG and EEG. So as I mentioned, in 2024, we acquired TMSI. That's a company that was focused on mostly EXG, so EEG and HD EMG. And now we combined the two companies, and we're also selling like combined FNIRS EEG devices. So a bit more about the founders. So one of the founders is Willy Collier. He's originally from Maastricht, so he's been there a lot of times. And the other one is Roland van den Burg. All right, let's first start with a bit of background about NIRS. So probably most of you are already familiar with EEG and the theory and how EEG works, but probably less familiar with what NIRS is and how you can use it to measure brain activity. So NIRS stands for near-infrared spectroscopy. And with NIRS, we measure changes in light absorption in a volume of tissue. And from these changes in light absorption, we can calculate changes in concentration of oxygenated and deoxygenated hemoglobin. Maybe sounds a bit complex now, but we go step by step through NIRS. So one of the principles that we use in NIRS is that human tissue is relatively transparent for light in the near-infrared region. And this is something that we can see if you take your phone and turn on the flashlight and put your finger on it. You see that most of the light gets absorbed by the skin, but the near-infrared red light and near-infrared light doesn't get absorbed that much. That's something that we can use in our technique. So as I mentioned, we have light in the visible spectrum, so light from 400 to 700 nanometers. So light from 400 to 600 gets mostly absorbed by the skin or tissue in the skin. And we use near-infrared light, and that's light with a wavelength between 700 and 900 nanometers. And how can we use this? So we are mainly interested in what happens with oxygenation in the blood, and therefore we look at hemoglobin, so which is the main transporter of oxygen in the blood. So we have two forms of hemoglobin. We have oxygenated hemoglobin and we have deoxygenated hemoglobin. So oxygenated is the one that carries oxygen, and deoxygenated is the one that doesn't carry oxygen. And what is a characteristic of the two is that they have a different absorption spectra. So what we can see here is that with the devices we transmit light into the tissue, and we use two different wavelengths. So we use 760 nanometers and 850 nanometers, and they both have a different absorption spectra, and that's what you can see in the figure. So at 760 nanometers, deoxygenated hemoglobin is the main absorber, and at 850 nanometers, oxygenated hemoglobin is the main absorber. So before 700 nanometers, biological tissue is the main absorber, and above water is the main absorber. So, and how can we go from these from this to actual concentration changes in oxygenated and deoxygenated hemoglobin? That's where we use this formula. It's called the modified Lambert-Beer law. So what we do, as I mentioned, we transmit light into the tissue. Light gets scattered, gets reflected, but in the end, part of it will end up at the receiver, and that's what we measure. So that's what we call the optical density, which is stated with OD. So that's what we measure. As I mentioned, we have the extinction coefficient. So as I mentioned, the oxygenated and deoxygenated hemoglobin have a different absorption spectra. We're interested in the concentration changes. So that's what we all want to have in the end. We know the distance between the transmitter and receiver. And since light is not transmitted, or it doesn't travel through the skin in like a straight line, it travels in more like a banana shape. So that's what you can see over here. We have to correct for this scattering in the tissue, and that's where we use the differential path length interval. So in the end, the only unknown that we have is the change in concentration, and so we can determine the changes in oxygenated and deoxygenated hemoglobin. So just a brief summary. So what we do is we measure changes in optical densities. From this, we can calculate the concentration changes of hemoglobin based on this modified Lambert-Beer law, and therefore we can calculate changes in oxygenated and deoxygenated hemoglobin, which tells you something about brain activity in a certain part of the brain. So what happens in the brain? So we have the hemodynamic response that we want to determine. So if a certain part of the brain gets more active, you see that there's neurometabolic coupling. So more activity means that there is a higher metabolic demand, and therefore you see that the part of the brain that gets activated uses more oxygenated hemoglobin. So the oxygenated hemoglobin will go down, and the deoxygenated hemoglobin will go up. But to correct for this, or because as a response, we see that blood vessels dilate to get more blood to that part of the brain that gets more active, and therefore the cerebral blood flow will increase, and therefore also the cerebral blood volume will increase. And if you take this all together, you see that in the end, all on the right, you see that in total the oxygenated hemoglobin in that part of the brain that's active will increase, and the deoxygenated hemoglobin will decrease. So the figure will look like this. So we have a few advantages in consideration of NIRS. So the advantages are that it's non-invasive. It's really easy to use. It's wearable and wireless. I will show you the devices in a bit so you can have an idea of how it looks. It's also compatible with other modalities, for example, EEG, which Konstantinos is going to tell you more about in a bit. We have a lot of many different fields of application. Consideration is that the hemodynamic response is quite slow. The hemodynamic response is relative. So what we do is we measure changes in concentration. So therefore, you first have to include a baseline. And then you can see if the oxygenated hemoglobin goes up or down compared to this baseline. And since we're measuring with a certain distance between transmitter and receiver, you measure half the depth of this distance deep. So you measure mostly the superficial structures and not the deep structures of the brain. So then the devices. So we offer some different devices to measure FNIRS, and that's what we call the Bright family. So we have some different devices. You see that they all differ a bit, but all uses like the same technique with the transmitters and receivers. So you see that the yellow ones are the transmitters, the blue ones are the receivers. And the main device that we offer is the Bright. So it has up to 27 channels. You can configure it and freely move the template across the brain, so you can change the location of it. So you can measure every part of the brain that you're interested in. We also offer a cheaper alternative of this device, the Bright device, and that's called the Bright Light. So what you can see is that it has fewer channels. It has up to 8 channels instead of 27. But you can also move it freely around the brain to measure the part of the brain that you're interested in. We also have designed 2 devices that mainly are designed to measure the frontal cortex, and those we have also in 2 different options, the Bright frontal and also a light version. And we have designed a device specifically meant to use for babies or children from 0 to 2 years. And we designed different optodes because it's more comfortable for the babies to wear. Yeah. So next to that, we also recently released a MediBrite. So that's a CE mark device. So it's can be used in clinical research. I would like to give the word to Konstantinos to tell you more about the EEG part.
Thank you very much. So I would like to present to you a little bit about the options that we offer to our customers for EEG and combined EEG FNIRS. So as you all may know, EEG offers some advantages. This is that EEG is non-invasive, it's portable and has high temporal resolution. It is widely accessible by the researchers, but also clinicians, because it's low cost and also can be combined with other modalities, for example, FNIRS or even MEG and fMRI. However, researchers need to bear in mind that the EEG has a limited spatial resolution. It is susceptible to artifacts, and it picks up weak signals from deep brain areas. So I would like to present to you the products that we offer as Artinis. So we mainly have two amplifiers. The first one is the Apex. This is a more lightweight amplifier. It can be used for portable applications. We're also going to demonstrate that after I finish my presentation. And the next one is the Saga. This is a more advanced amplifier. It offers more options to the researchers. And then we also offer a variety of EEG head caps. On the left, the first one you can see is a pin electrode head cap, where you use gel in order to improve the conductivity with the scalp. Then we have a ring electrode head cap. This you can see on top. And then we also offer response-based electrodes that use water in order to conduct electrical activity. A bit more details about the Apex. So it is a lightweight and portable amplifier. It picks up high quality EEG signals, and it can be used for mobile but also stationary applications. It has an onboard memory space, which also helps. For example, if you don't have a laptop close to you, you can save the data there, and then when you go back to the lab, you can put them in your PC and view them and export them. It comes with a 24 or 32 EEG channels, has a trigger connector, and it has a high sampling rate to capture a full band EEG signal. And it can be combined with our FNIRS products, as we will also see in the demo afterwards. Then we offer the Saga. Saga is a more versatile amplifier that can offer additional options to the researchers. For example, it can record both EEG but also high density EMG signals, so also muscle activity. It also can extend to 64 EEG channels in that case. And it can be combined with external sensors, for example, for galvanic sensor, skin response, 3D accelerometers, or oxygen saturation. And it is also compatible with our FNIRS products. Now I would like to focus a bit more on the last part of the presentation about how we can combine EEG and FNIRS. And I think this is a really interesting thing to see, and because it's also going to affect how the field evolves in the future. So as a company, together with the amplifiers I showed before, the EEG amplifiers, you are able to combine it with the devices that Sam showed before. This is a Bright FNIRS device, and we offer two options. The first option is to place the optodes, which are from the FNIRS, and the EEG pin electrodes on the same holder. In that case, you can measure almost from the same location both signals. Here you can see, for example, an optode and an EEG electrode next to it. We're also going to demonstrate that in a bit. And then we also have a bit more classic, let's say, traditional approach, which is to actually place the electrodes and the optodes next to each other. So for example, in that case, you would like to place your electrodes in the 1020 configuration. Also, we collaborate with external partners to allow our FNIRS devices to be combined with their EEG amplifiers or head caps or electrodes. One of them is Brain Products. Also, A&T Neuro and Biosemi and GTech are other examples that we offer combined solutions with. And I would like to take a moment to also show the benefits and also considerations regarding combining the EEG FNIRS. Well, the most big advantage is that it gives a more complete picture of the brain. It has the fast EEG signals, but also the slower signals from the FNIRS that picks up the blood oxygenation changes. Both devices are quite similar in the sense that they are portable and low cost, and they can be used for naturalistic real-world experiments. And they can be applied to diverse populations from babies to elderly people. However, there are some technical challenges that we try also as a company to actively tackle and provide the best solutions to the customers. The first one is the synchronizing of the EEG with the FNIRS signals. The EEG signals are a bit more fast, the FNIRS are a bit slower, so that can make it hard, how to design your experiment and also how to synchronize them. In that case, we offer hardware, the so-called PortaSync device, which can synchronize with buttons, for example, your signals, but also you can stream your signals in the lab streaming layer, and you can synchronize them there. Another thing is that our scalp, as you know, has a limited space, so it cannot take too many number of sensors if you use both modalities. However, as I showed earlier, we have these combined holders, which helps with that because you almost place at the same location, both the FNIRS and the EEG. And lastly, FNIRS may introduce artifacts to the EEG signal. This is something that we actually right now are investigating, and we'll soon make a blog post about it and share it with our customers. Regarding the application areas, there is a wide variety from the clinical practice to a classic cognitive neuroscience experiments. Since it's portable, it's really popular in sports science, and also in hyperscanning experiments where you can study the social interaction between two different subjects or more subjects. And can also be applied for developmental studies, but also for gut and brain interaction, which is closer to your field. And I just wanted to make a slide to close with how can EEG FNIRS benefit the field of gut and brain interaction. I read through some papers and I see that there is increased interest for more personalized research in naturalistic settings. And I think there the combined EEG and FNIRS can really give advantages, offer their advantages. Since they are portable, so they can allow studies in home settings, for example. They are affordable, so they can allow for larger clinical studies, and they fit all head sizes. And closing, I just wanted to say that I didn't find many papers about combining EEG FNIRS, but I think soon more and more people will use it. And I would be really curious to see how we can get a more complete image from the brain using such modalities. Yeah, thank you very much.
Perfect. Yeah, then it's time for us to do the more fun part, to show you the devices, how it works. So first we will set up the cap with the combined holders. So casinos have to prepare to gel me and how it goes. Now we'll talk you through the software. Can show you how the near signal, what it looks like, how the EEG signal, what that looks like, and can show you some devices and I think you're interested in. So
yeah, if you want to take some questions,
you can do it during the demo as well. can do it also now if there are.
Yeah, so I would
questions
say
already.
because we will start setting up slowly. That takes a little bit of time, so it would be nice to actually take questions at the same time.
Yeah.
And you can start, and I can answer maybe some questions.
Yeah, definitely.
I'm only gonna gel the subject. I'm not gonna.
Yeah, maybe one question that I had.
Yes.
One of the final slides when you say that the same FNIRS device, for example, could be used for for other organs, right? The previous one, you had liver, kidney, and so on.
Here.
I mean that even the same sort of headband as at the column left can be used then just placed on the chests of the participant, or there is also like different shapes.
Yeah. So I think would you like to take that question?
Sorry, I was preparing.
So what I know is that such headband, it is used only for brain recordings. It's set up so you can place it on your head. So we don't use that for studying, for example, the liver, the kidney.
Yeah, there are modifications that we can do. So we can also use different wavelengths. I know that for liver clearance, they also use ICG. It's not sure what it's called, but they insert kind of fluid to see how fast the liver breaks down certain fluids to see the liver function. But you can also use it to see what happens with the oxygenation in these tissues. Yeah. But some tissues, for example, the liver or the stomach lies a bit deeper, and you're limited to a certain range how deep we can measure. So sometimes that presents a bit of difficulty, but we have options to customize devices for certain needs. So that's there are definitely possibilities to do that.
Yeah, because what I had in mind is that okay, now you have the FNIRS EEG combined, but it could also be the possibility of having EGG and the FNIRS on the sort of the stomach, for example, or in your intestine.
Yeah. So with the Saga that we have. So here you can insert like the head caps, but we also have the possibilities to use different channels. So we also have auxiliary and bipolar channels. So in that case, you can also add some snap-on electrodes to measure, for example, EGG on the stomach the same time. So there are definitely possibilities to do these kinds of experiments. Yeah.
Yeah. So I will be with Saga, which is an EEG amplifier. What about the FNIRS? The possibility for such.
Yeah, so in that case, we offer a device of a partner that we work with that's called the BitBrain, and that can be used to, for example, measure EMG, ECG, but also EGG. But then you're also using like the bipolar or snap-on electrodes for. So there are certainly possibilities.
Any other questions? Yes, please.
Different is the BettyBright from the other models? Because you mentioned that it's more suitable for clinical research, but what modifications did you make to make it more suitable for research?
That's a really good question. I know that it just has the same functionalities. It works actually quite the same. It's only that it has a CE mark. So we took that in account. It has a bit longer optode, so the fibers are a bit longer. And you have the option to choose from more flat. So what you can see, I can show you later. But these have more like a sharper tip, for example. And there you have the option to choose for more flat for subjects who are more skeptical for these kind of things. So therefore you have designed different kind of optodes and transmitters to make it more suitable for clinical populations.
And of course, pass like all the tests that you want for a CE mark, because there might be also things related to the signals. What happens if the device has an issue? Can it harm the participant? So it needs to pass some tests. But yeah. Yes, please.
How does it work? I mean, in general, how can you get a good signal avoiding any artifacts due to contact resistance between the and your electromagnet? There are many factors that may interfere with the EEG, but I'm talking about the electrical signal.
Okay, so you asking a question when you combine both modalities, or
producing a reliable contact between your electrodes and the tissue you are.
Okay, when you only use EEG, not
only
both modalities. Yeah, so like the first, I would say step starts from, as an experimenter, you start from the experiment. You need to put enough gel. I think this is something that many people say.
Using an ionic gel. A gel.
Yes, the gel, which helps for the conductivity. And then I guess then you move to the data analysis. Now, for us as a company, what we try to provide is, for example, our electrodes have active shielding on the cable, which basically helps to get better signals and not get affected from, for example, the 50 hertz interference so much. So we run a lot of tests in order to improve that and give to the customer the best solution. So I would say it's a bit from both sides.
But are you trying to develop something which is gel-free, so you don't depend?
Yeah, so we do have the water-based electrodes, which you just need to soak them in water with saline to just get high enough conductance, and then you can measure. We do not offer currently dry electrodes. There's been in the market such electrodes, but there's still consideration to how good signal you get. It's easy to apply them, but the traditional, I think, ways to put the gel, which we know, we trust it. Yeah.
Yeah,
And
I'm just considering
Yeah.
the chance to have a wearable electronics which you don't want to check.
Yeah, I understand. Yeah.
And maybe two a little bit related questions, maybe. I was wondering, related to signal, what we have like with transcranial Doppler, with ultrasound, we have major problems with older adults with the skull calcification that it is with the chill signal. Do you have like similar problems with this device? That like with calcification of the skull at older age, especially ladies, that you have problems with the signal, or is that not the case?
So you're asking for FNIRS, right? Not
Yeah.
for EG in that case.
Okay,
For FNIRS, it's not a problem. So it's just transparent. It just goes through.
okay.
The only thing is that they have a bit less dense skin.
Yeah.
But what we offer, the Bright has, it has what we call automatic gain control. So it changes light intensity of the transmitters based on, for example, if people have darker hair or darker skin, or so that automatically adjusts the intensity based on the certain subject. And in that case, it accounts for these certain factors to have the most optimal signal quality.
And how does the device deal with movement artifacts? For example, can you use it in real life? For example, if you put people on a treadmill, can you measure signals when people are, for example, walking?
Yeah,
Or do
definitely.
you see like major artifacts?
You see, but we have certain algorithms that can be used to filter out these movement artifacts. So
Okay.
that's definitely done a lot
You,
during dynamic movements. Yeah.
Wow.
yeah, sorry for interrupting. You also have an IMU sensor.
Yeah,
On
there's also,
the
yeah. So
FNIRS.
in the control unit, so here we can see the Bright that I talked with you about. So there's also an integrated IMU, so with accelerometer and gyroscope, and this data can be used in the algorithm to correct for these motion artifacts.
Yeah, that would be post-processing, post
Yeah.
acquisition.
Yeah.
But also for the EEG part, based on the scalp, I think the scalp is really important for EEG signals because we know that it basically blurs the signal depending on the scalp. You have the volume conduction, as it is called. And during analysis, you also need to model the scalp as good as possible if you, for example, want to do source reconstruction. So in that case, I definitely see the scalp being a bit of an issue, but it can be solved, for example, in fMRI of the subject. So you can basically totally show how the EEG signals flow, and then you can understand from where it comes from.
Another thing to come back to your question about the calcification, and we also, so now we have channels that are classic 30 millimeters. We also have the possibility to have short separation channels. Those are like 10 millimeters, 8 millimeters apart. And this is what we use to correct for the superficial layers like the skin and the skull. So what you basically does is subtract the signal of the short channels from the larger channels, so that you only have the signal that you're interested in left.
Maybe regarding the source reconstruction, I'm wondering if combining EEG with the FNIRS could help also the spatial source reconstruction of EEG, because of course they measure very different things. No, one is the physiological oxygenated blood signal, and the other one is the current of the neurons. No, so do you provide any algorithm or technique or something of this nature to help the EEG spatial source reconstruction from the FNIRS signals? Or if you implement something at the hardware level. I mean, I'm thinking about the disposition of the electrodes or something like that, or you analyze them separately.
Yeah, I could take that question. So we do not provide an algorithm for that. I also personally do not have any knowledge about how you can use both modalities in order to improve the source reconstruction, because in the end they come from two different sources. Like the one comes from the blood oxygenation and the other comes from the electrical activity. So it physiologically, neurophysiologically is a different type of signal. So also like thinking right now about it, I don't see how the one modality would help the other modality to improve, for example, the source reconstruction. I think the idea is that at the same time, you can measure both things, and then you can do the tricks that we already do with EEG and the tricks we already do with the FNIRS to say, for example, we have these different measurements, but at the same time, not to help each other improve their data analysis.
Questions for now? Otherwise, we'll just continue with the demo and see if there are other questions will rise up. Yeah.
Okay.
Let's go.
So you need to share your screen, right?
Yeah.
So you're going to share
yeah.
the software.
Yeah.
I can do it if you want as you work with the hardware.
But I can also share it. Now, if you have to stop sharing your screen.
I did stop mine. But most of the people here work with EEG, right? That's the predominant modality.
Software, right? So what we do offer, so we have separate softwares for EEG and FNIRS. So for FNIRS, we have what we call Bright Connect. So as you can see, it's not the most fancy looking software, this one. This is called OxySoft, but we have a software, Bright Connect. That's our acquisition software that looks a bit more fancy. For the EEG part, we have a user application as well, which looks a bit more fancy, but this is our software OxySoft, in which we can integrate both FNIRS and EEG. It has a lot more functionalities to do some analysis like filtering, some block averaging, these kind of things. So therefore, we would like to show this software to you today to show that we can measure combined FNIRS EEG, and also shows you some functionalities of the software.
All right.
Right, ready?
Would you like me to put the head cap on you?
No, I can do it myself if you will just install the electrodes.
Yeah. That's good for me because I always struggle to put the head up.
All right.
So we have different head cap sizes, right? Depending on the head.
Yeah.
Now you're using a
Size L.
L. It's a big head, and it still feels like it's very tight, no?
Yeah, so that's what you want. You want the optodes and transmitters to have good contact with the skin. Therefore, you would like to have the right size for the right participant.
Yeah.
I think we should.
I'm just gonna, if you could hold that. So now we have placed the FNIRS device. Here is the control unit in the back. And then the channels, the optodes as we call them, have been placed in their holes, in the covering for now only the frontal lobe. And now I'm gonna place on these holders, which are the so-called combined holders, the EEG electrodes, which come from the control unit or amplifier, as we call it, the apex.
Place the reference.
Yeah. So the apex, you hold the apex?
I have it in my pocket.
Yeah, you can put it in your pocket.
Also to show that it's really wearable and that you can take it anywhere else during dynamic experiments.
So we have already placed 3 combined holders. So I will place 3 electrodes in the front, and I'm placing one more combined holder in the back, which is we're going to use as the reference for our measurement. Let's see if I can fit that in. Okay, so it's pretty easy to put the holders there. Basically have a bit of space. So this has like a hole here, and then you just clip it in. So in that case, I'm gonna place the FP, as we can see in the cap. We have the FP2, FPZ, and FP1 electrodes. I'm just gonna place them in the combined holders. So I start with the FP1. In that case, this would go FP1, so it goes here. So the idea is that it's like a pin electrode. I'm just gonna plug it in and then turn it in order to lock it on the combine holder. Or shall I gel it first? What do you think is better?
You can also gel it first.
Yeah, so I think I will gel it first just to make sure we have good signals.
Yeah, we're not going to do all channels today. Because you're probably familiar with to gel every
So this is the
channel for EG. It takes some time.
I'm gonna put enough. I'm gonna make him.
So for FNIRs, it's a lot easier to get good signal quality in quite a short amount of time.
What you can also do with this syringe is that you can move a little bit the hair of the participant. There is a little bit of space just to get better contact with the scalp. Right now, we have a nice forehead here, so I think this is not going to be necessary. You feel it? Because
Yeah, definitely.
yeah.
Have a recommendation for having a good FNIR signal?
Sorry?
What are the recommendations to have a good FNIR signal?
Yeah, so you have to have good contact with the scalp, preferably, because the hair, of course, since it's some people have darker hair than others, but hair absorbs part of the light. So we have like a hair removal tool to move the hair away and to make sure that you have good contact with the scalp. That's actually the only thing you can take into account, and that you choose the right head cap. No.
You felt the gel in the front?
The other two not that much.
Okay. This one has the thing. And then okay, let's hope for a good EG signal.
Right. Then in the meantime, I will show the software and
Yeah.
Okay. So this is what the software looks like. So we have a screen. I just created a measurement. We can continue to the next step. So here are the devices we are going to connect. As Konstantinos also already mentioned, we also have like a synchronization device. Oh yeah, sorry. Maybe a bit too early.
Maybe it wasn't a good idea to put it on the frontal.
Yeah. Meantime, I will connect the devices.
And just put it in, turn it, it locks. Okay. And we have, let's see, FP1. Turn that a little bit, come inside the hole. All right, the last one. Okay, I made it. And this is the reference one. But the idea in general is now we didn't do it so well, so you should turn the holders to be pointing backwards so you can have the cabling like coming from the back. Now I have them pointing downwards, so it's not very good for the subject. It's just the cables going from the face, so can be pulled a bit. But anyway.
In the meantime, I can tell you a bit more about the software. So what you can see over here is that you have the option to choose different channels. So you have different devices. They all come with different channels. So for example, you can choose certain configurations you can use on certain parts of the brain, depending on your specific needs, requirements, and what you would like to measure. So for now, we're using the bright light, so the one with only 8 channels. So we can select it here and select the 8-channel template that we have over here. And we can continue to the next step. So in here, you can select the sample frequency. So with FNIRs, you're measuring, since the hemodynamic response is quite slow, you don't need a really high sampling rate. But of course, for EG, you can. So therefore, we have a higher sampling rate here. It's automatically synchronized in the software. So the timestamps are automatically synchronized by the software, both the EEG and the FNIRS. Continue. So now, as I mentioned, we have like the automatic intensity, the gain control. So now it's performing just the setting adjustment based on my type of skin and hair, on what the ideal signal, the light intensity is. Takes a while. So now we're seeing all of the nearest channels over here. We have some EEG channels here. You see that we don't have a really clear signal yet.
Yeah, it's probably on me.
So in the meantime, we can talk a bit about the nearest channels. I can make them a bit larger. Can you see it already? Yeah, it's quite big. So now we see like a bit of a wavy pattern. Do you have any idea what that could be, what we see in the signal right now? No, it's not the lights, because we have like a really dark neoprene head cap, so that helps blocking out all of the light of the environment. But what we see in the pattern, that's the heartbeat. So since we have a pulsative signal, you see that the oxygenated hemoglobin gets increased after every heartbeat. You can show that here you have a nice clear signal.
So what is the red and what is the blue?
So in red we have the oxygenated hemoglobin, and in blue we have the deoxygenated hemoglobin. So for example, now we have placed it on the prefrontal cortex. It's a bit hard to do a live experiment now, but if later you're interested, we can also set it up on you, for example, and we can do an example with the motor cortex, for example, a finger tapping task. And if you start finger tapping, for example, you see on the motor cortex that you have like the nice hemodynamic response as I told you about. So if anyone's interested in trying, feel free to come by our booth to try the device yourself. That's going to show you now. As I mentioned, we also have the IMU integrated. So I can also add the data of the, so I can also EEG channels. So you can just select all the channels that you're interested in. Most commonly you're using all of the channels, but for now we're using only 3, so you can just select them. So here you can see that we also have the accelerometer and gyroscope data. So I can select those to be shown on the screen as well. So now in here you can see that if I move my head, that you see that there's, that the signal is measured. So we also have a lot of export options. So if you go to measurement, I would first have to start in the visual recording. Then you have the option to export. For example, we have the oxy files, how we call it. We have the possibility to go to Excel, to text, to CSV, or other commonly used files in EEG. We also have the snare file, the .snare file, that's commonly used in by all kind of companies that are using FNIRS, to have like the same format, which you can then load in different toolboxes, different analysis toolboxes, or for the algorithms that I told you about for the motion correction or other toolboxes. We also have like some Oxysoft to MATLAB scripts to load it in to customize your own scripts and get the data out of it that you're interested in. Any questions so far?
And you can also build your experiment in there, or you simply get trigger codes via the amplifier.
Yeah, you can also use LSL for that. So you have different programs to build like your own experiments, for example, Psychopar or something, to design all kinds of tasks, and then you can get automatically events inserted into the data by this other program. So we also have some options to do some calculations, to do some filtering. So what you can see over here is that you can apply some filters. So we have different options like moving average, bandpass, moving Gaussian. So if you apply that, you can see that the near signal is filtered and the heartbeat, for example, gets filtered out. But also for EEG, you can use all kind of filters, also during live measurements if you prefer. So there's a lot of possibilities that you can do with the Oxysoft software. Any additions
No, nothing.
so far?
I'm a little bit disappointed with the EEG drilling and the placement.
Yeah.
You have placed the ground electrode, right?
Yeah.
Yeah, so like also we have this hand band sweat.
Yeah, I've wetted
Yeah.
it
So
before.
you can put it in saline water to increase the conductivity, and then you just place the ground electrode on it. But it's like a basic thing for EEG signals to help in the right range. I see like a bit being outside of the range, we won't maybe.
Yeah, if you're interested in seeing good
conductivity
EEG signal, we can also try it later
Yeah.
ourselves. Let's see if we can show you the EEG signal. If there are no other questions, I think can end the workshop here. And thank you for your attention. And if there are any questions, just feel free to come by and can answer all of your questions and tell you more about the devices, the possibilities, and whatever you're interested in. Thank you.